So good morning and welcome. My name is Paul Hann, and I'm the Acting Chief of the TMDL section at the California State Water Resources Control Board. Before we get too far into this, I want to provide some general information to help with managing expectations. As you know, the Water Board is developing nutrient objectives for the state's surface waters. We initiated this effort back in 2000, focused first on lakes and streams. The technical work has been completed, and the approach has been used successfully to address nutrient impairments within a number of specific water bodies. We're now working on developing the approach as a statewide policy that would apply to all lakes and streams. The draft policy document will be released for public review sometime in the second half of 2013. However, I want to be clear that today's webinar will not discuss either policy or technical issues related to nutrient control within lakes and streams. What this webinar will focus on today is on technical development work within enclosed bays and estuaries. We began this work in 2008 and are still working on the science to support future policy development. We don't expect that the policy development work will begin in earnest until 2015 at the earliest. For this reason, I'd like to ask that you focus your questions and comments on the technical development work you hear today and leave the policy questions for another time. Next slide. So to introduce Dr. Martha Sutula, who will guide us through the rest of today's presentation. Martha is the head of the Biogeochemistry Department at the Southern California Coastal Water Research Project, also known as SWRCB, where she sees projects related to eutrophication and harmful algal blooms in streams, estuaries, and nearshore waters. SWRCB is the technical lead on science support to the Water Board for nutrient objectives. Martha?
I really appreciate everyone attending today. It's a pleasure to be able to present some of the science that we've been working on over the last couple of years to support Estuarine Nutrient Numeric Endpoint framework. What we have in store for you folks today is basically, I'd like to start out by just refresh your memory if you've already heard about this, or presenting it to you for the first time if you haven't. Then try an overview of the State Water Board's conceptual approach to nutrient objectives. Really important for context for the science that you're going to be hearing about today. And then I'll talk about specifically why macroalgae, why are we thinking about numeric endpoints for macroalgae. The presentation will be followed up by Dr. Peggy Fong from the University of UCLA, who's going to actually spend some time talking and giving you an overview of what algae is an indicator that we would want to consider for objectives, and she'll go into a little bit of the ecology of macroalgal blooms and estuaries. And then once we wrap that part of the presentation up, what we're going to be doing is making presentations on the studies that were done to support the macroalgal anemone, intertidal flats and unvegetated subtidal habitat. We'll kick off with Dr. Lauri Green, who was a postdoc working on this project, presenting the effects of macroalgal blooms on benthic fauna. These are field experiments that Lauri did, and then followed up with the results of a field survey that we did using sediment profile imagery. Basically, both of those studies targeted at trying to understand what are the thresholds associated with macroalgal blooms in these habitat types. And then I'm going to wrap it up by giving you a synthesis of what we learned and then talk about next steps. Okay, so really helpful to understand about nutrients is that the approach for setting nutrient objectives is really quite distinct from that for traditional contaminants. And what's really important to understand is that nutrients are required to support life, but there is clearly some point in which there is too much of it going into our streams and our other bodies, but there's how much too much, and sometimes that's a nuance. Also really helpful to understand is that unlike the paradigm that we would typically use for toxicity for other contaminants, for example, toxicity is really the endpoint of interest for nutrients. We have some severe effect occurring at much lower levels. And in addition, whereas the focus for traditional contaminants is refocusing on the surface water column nutrient concentrations use nutrients in the water column can often give you a false negative or false positive. For example, this estuary shown in this picture is meeting basin plan objectives in Region 9, the San Diego Regional Board's basin objectives, but really, there's a clear problem with respect to overgrowth of algae, and so this is the reason why for nutrients that we really need a different approach. California's approach to nutrient objectives is basically the idea starts with an objective, coming up with numeric guidance that essentially helps you translate that narrative objective, and that guidance is what's being coined as Nutrient Numeric Endpoint, or NNE. So if you hear about the NNE, this is essentially the numeric guidance that would help to translate a narrative objective for nutrients and biostimulatory objectives. And then behind the NNE, there are two basic tenets. The first is that the diagnosis is being based on response indicators. And so this is what I will refer to this in this talk as the NNE assessment framework. And what you're doing is you're assessing the effects of nutrient over-enrichment, which is what we call eutrophication, and any adverse effects of nutrients. And when possible, we also like to be able to use multiple lines of evidence for a more robust diagnosis. The other sort of basic tenet of the NNE is that you need, going to make a diagnosis based on response indicators, then you're going to need some sort of model to link those response indicators back to nutrients and other factors that you can measure, for example, hydrology or even those that you can't, like climate, to be really make sure that you're getting the right nutrient number. And the models come in a variety of different flavors. They can be empirical, statistical models. They can be dynamic simulation models. But using models then will be a key way to get to what we would need to manage, which is nutrients. I think the advantage of the NNE concept and why this is important is that even lots of water bodies where you may have very similar nutrient loads, you have some very site-specific factors, what I would call cofactors, within that water body that are essentially mitigating or changing that water body's response to nutrients. And so within that same water body that has, or similar water body types that have similar loads coming into it of nutrients, you can have some very different symptoms of eutrophication. And so it's important to be able to take into account these specific cofactors or environmental variables and you set the numbers for nutrients. And so what we're doing then is we're picking when we think of assessing eutrophication and setting a number, we're picking among the different types of ecological response indicators that essentially are the symptoms of eutrophication. And then the advantage is that these are much more closely linked to the ecological endpoints of concern. These are the things that we measure our water bodies for, much more directly linked to beneficial uses. So, an example of how this plays out in streams, and Paul had mentioned that the science supporting the NNE for freshwater streams and lakes is essentially complete, and they're off working on the policy development. And so this is an example for streams. Essentially, what we're using is a combination of benthic algal biomass, pH, and dissolved oxygen. And just as an example for benthic algal biomass, what we're essentially doing is setting these thresholds beneficial uses, and that is essentially how you would assess whether or not the water body is having a problem. And you would be using these target response indicator endpoints as a way of calculating what nutrient numbers would also go into permits, for example, or for TMDLs. So coming back to this concept, so you start out with response indicator thresholds, then you're going to use models as a way of getting back to those nutrient numbers. And so I mentioned earlier, these models can be in the form of empirical statistical based on field data. You can develop more spreadsheet models based on mechanistic relationships or numerical models. And it's clear that depending on the model type and what you can use, there's a variety of different considerations, including data requirements and cost, and also what you want to use those models for. Right now, the state is offering a set of spreadsheet models are scoping tools to help with the application of the NNE in lakes and streams, and this is part of the NNE package. So that's sort of a broad overview of what the NNE is. And so I think when you start to think about how this concept translates to estuaries, it's actually the same conceptual approach, but the indicators and the thresholds and what appropriate models will be for estuaries will differ from what we see for lakes and streams. For approaching the NNE effort is that we have we divide work into two phases. Phase one is the development of these NNE assessment frameworks, and then phase two would be development of working on the response models. And this phase one, essentially the process is, number one, to identify the indicators. What are the appropriate indicators? Second step would be to construct these assessment frameworks, and in the process, identify what the appropriate thresholds are. And there's really two elements that go into this. The scientist work is to essentially and synthesize what data are available for decision making, and the policy makers make those decisions informed by the stakeholders. So basically soliciting stakeholder opinion about what policy decisions they're proposing to make. We've already completed some of these steps for the Estuarine NNE. We've evaluated candidate indicators vis-a-vis a very explicit set of review criteria. We've looked at several studies that are available to establish thresholds when they're available, and we summarize that information. And all this information is in a report that was published last year and is available on the SWRC website. And I'll draw your attention to a chapter that was authored by Fong and Lauri Green, who are on this call with us today, as well as Rachel Kennison, specific macroalgae. So this is an overview of what makes macroalgae a good indicator. Is it a good indicator? Goes into the so there's an extensive chapter available for you folks to review that gives you a lot of background on that, and I would invite you to read that. About estuaries, it's really helpful to take a big step back and recognize that within the state of California, we have lots of different types of estuaries, and that plays into how we would implement the NNE. From a large perspective, we have three sort of major classes or geoforms that we call them, and there's enclosed bays, lagoon, river mouth estuaries. The enclosed bays are really what's represented as the largest proportion of habitat in terms of percent of area in the state. The lagoon river mouths, as a percent, much smaller. They probably represent about 10% of the state by area, but they're actually some of the most numerous types of habitats that we have. Generally, what happens is that these lagoons and mouths can be aggregated into sort of a superclass of estuaries called bar-built estuaries. And the significant thing about these estuaries, they're very typical of Mediterranean climates, that their mouths close, sometimes seasonally, sometimes they're closed for very long periods of time and only open during large storms that have enough to break open this sand barrier. But what happens then is that this controls the interaction with the tides, and so you have some that, because their position are always open, we call them perennially tidal. Some are open for parts of the year; those would be intermittent. And then we have some estuaries that are ephemeral, that are almost always closed and only open during large storms. And we need to take into account this classification as we think about indicators and how this plays out. This into account is the habitat type. So within these estuaries typically have some form of these four types of habitat: flats, seagrass, or some type of submerged aquatic vegetation, unvegetated subtidal habitat. When we think about what we assess in the NNE, we only focus on those three indicators. We are at this point excluding emergent marsh because that appears, at least from our understanding, to be the least sensitive of the habitat types when we think about assessing eutrophication. So these are the recommended indicators that resulted from that 2011 report, broken up by subtidal, intertidal flats, and shallow subtidal and seagrass. what we're basically looking at is a mix of dissolved oxygen plus primary producers, things like phytoplankton, macroalgae, and the effect those two types of things can have on, for example, seagrass, as well, things like epiphytes. So the idea is that depending on the specific habitat type, indicators are more or less the same, phytoplankton and macroalgae, but how to assess them would be different depending on the type of estuary. Estuaries, what's also really important to understand is that when that inlet closes, the status really controls what prime producers are dominant and what they look like. When it's open, they look like a lot of other perennially tidal estuaries. For example, they're typically dominated by intertidal flats. You can have a combination of macroalgae and benthic diatoms. But when they close, it looks more like subtidal habitat. There's very little of what you would call a mud flat or intertidal flat. And your macroalgae looks much more like sort of a rafting variety of macroalgae. You can submerged aquatic vegetation. Rupia is very thin, and you can also have phytoplankton. When we think about classification, what happens then is that we boil it back down to there are more elaborate classification boils down into something quite simple, which is estuary in an open state, so it's circulating with the ocean, or it's closed, in which you have a sandbar that's blocking it. And then we use this more simple classification to think about what indicators and what thresholds we would need to develop. Then think about it this way: we would, both open estuaries and closed estuaries, when they're in that state, share both dissolved oxygen and primary producers as type of indicators. But then the thresholds could be potentially different. If we're using an open estuary versus a closed, guidance for how we measure it and how we would use data to make an assessment would also be really different. And so this is how we're conceptualizing it. I'm going to put aside dissolved oxygen. That's something else that we're working on, and we're not discussing it all today. And really, then we're thinking about primary producers. What we're talking about developing is a phytoplankton NNE, which has components in vegetated subtidal seagrass and subtidal for closed estuaries. And macroalgae, which we are talking about today, that has components in seagrass, intertidal and shallow subtidal, vegetated habitat, and subtidal for closed estuaries. So this, when I talk about a macroalgal NNE, it actually has three components, and the slide we're presenting today is really representing one component, which is this intertidal and shallow subtidal unvegetated habitat. So in terms of studies that are ongoing or have been completed to support this macroalgal NNE, there are three types of studies going on: field experiments and surveys of effects of macroalgae on intertidal flats. That work is complete. This is what we are presenting to you folks today. There's also field experiments and surveys going on for seagrass habitat. That work is in progress, and that work is also being led by Dr. Peggy Fong and her students. And then finally, we have another study, which is a field in closed estuaries, documenting the natural background abundances of macroalgae and phytoplankton in closed estuaries, and that work is beginning next year. What we are presenting to you today is really just the results of this first of three studies, so it's helpful to understand the context in which we would be using this data. The last thing I wanted to present to you folks today is an understanding of how we are using the language of talking about thresholds in this particular presentation. So it's really helpful for us to all start on the same page. When we wanted start looking for the numeric endpoints for these response indicators, what we're talking about is finding the thresholds that begin to see some sort of environmental change in the system. So what this graph is showing is ecological condition on the y-axis, the stressor on the x-axis. And in this particular circumstance, what we're thinking about in terms of the stressor is not nutrient nutrients per se, it's actually that response indicator. So if we're looking at macroalgae would be the response indicator driving some change in environmental conditions. Then when we're thinking about for these thresholds, we're looking for some understanding, because in this case, macroalgae is a natural part of the system. What would be a reference range of macroalgae in which you would that's sort of what through variability you would expect to have. You have some point where you have so much macroalgae that you start to see changes in some indicator of ecological condition. So there's a first point at which you start to see some sort of environmental change, and then at the outer range of macroalgal biomass, at a point you really start to see some essentially crashing out, or the actual point of termination of that indicator, when it's essentially got so bad it maybe has disappeared entirely, or is no longer recoverable. And when we think about these thresholds, these are resistance and exhaustion thresholds, and also really helpful to understand in terms of what we can actually measure. Or, if we do experiments, and Lauri Green will be presenting to you folks today experiments, those experiments essentially reflect benchmarks. Benchmarks can reflect where you're documenting some adverse effect level at some specific set of experimental conditions. For example, certain areas of certain grain size, certain water conditions. And it's also possible to identify levels or benchmarks in which no effect has occurred, or in some cases even enhancement in that environmental variable. And so these thresholds, when we think of them, they can be theoretical constructs, but we can also use statistics when we have appropriate data to statistically identify those thresholds. And so it's important to start out with this control model for what we're trying to do today to identify these thresholds. And we're approaching this is, it's important to understand that in terms of identifying numeric endpoints, there are actually several different ways in which we can arrive at regulatory or policy decisions. There's best professional judgment. There's use of characterizing what's referenced for the system. But one of the ways, and maybe the ways that we like to be able to rely on when we can, is actually documenting the relationship between the dose of that stressor and the response. And so that's something that we've been able to do fairly well for macroalgae, and this is what we're going to be presenting to you folks today. Okay, so with that, what I'd like to do is introduce you folks Dr. Peggy, who's going to be talking about why macroalgae is a good indicator of eutrophication. It's been my great pleasure to work with Peggy Fong. She's a full professor and a vice chair of UCLA's Department of Ecology and Evolutionary Biology. Her research lies at the intersection of community and ecosystem ecology, with an overarching goal of using those results to inform the conservation of marine ecosystems and communities. She studies the effects of climate change, nutrient enrichment, accelerating human use of natural resources on many critical habitats, including coastal estuaries and lagoons, coral reefs, kelp forests, and seagrass beds. And she is one of the world's foremost experts in macroalgae, so it's been a great resource and a pleasure to have her on board working with us on the project. And with that, what I'd like to do is turn it over to Peggy. Thanks.
Hello, everybody. I'm sorry, you're on mute, so I have to assume you can hear me. Okay, here we go. My job today is really easy. I just want to remind you again of some of the things we have in that really large team that Martha showed you the cover earlier. And basically, I want to tell you a little bit about ephemeral macroalgae and they bloom and how they bloom. So the first thing I want to talk about a little bit is the taxonomy. You come in three flavors, I like to call them, and they really are colors. They're red, green, and brown based on their pigments. And I have the red and green highlighted here because those are the ones that generally form blooms. Mostly in Southern California, the coast. As you move north, it can be a mixture of the reds and the greens. But the important thing to stress is something Martha said earlier that I can't stress too much. macroalgae are natural parts of ecosystems. They support really vital ecological functions across all ecosystems. They are the base of the food chain. They provide oxygen, food, shelter for other organisms. But of course, like all good things, sometimes a little bit good and too much, a little bit is too much. Macroalgae come in a variety of forms. They have extremely diverse morphologies. You and Kelly know about the giant kelp. So things range from single-celled macroalgae from simple sheets all the way up to meters-long kelps. Things that form bloom species overall, in general, tend to have very simple physi, or that's the algae word for the body of the organism. So they're very simple forms, simple sheets, so sort of morphologies. They often undergo changes in habitat usage through different life stages. So start off attached to the benthos, the bottom of the ocean, or the estuary, and then they often, as they get larger, detach and form these large floating rafts that Mark showed you some nice pictures of, and you'll see more pictures of. So macroalgae are ubiquitous. They're found throughout many different habitats and estuaries, all of the ones Martha talked about. So on the surface of flats, as epiphytes on seagrass, and as large floating mats, usually in deeper brackish areas and deep water enclosed bays. There's what's going on in these slides, and what I really want you to concentrate on here is simply these are two different forms of green algae, one on the left, one on the right, and these are just nutrient uptake rates. And if you look at the circle in red, the 100 and the 150, that's how many micromoles of nitrogen they can take up dry weight per hour. And just to put this in perspective, this is often an order of magnitude or 10 times higher than the concentration of nitrogen you find in seawater. So these things can take nutrients and scrub them down out of seawater very, very rapidly. And that rapid uptake results in rapid growth. Other sorts of interesting twists is that having a really high nitrogen supply also enhances their ability to tolerate environmental extremes. Now remember, macroalgae marine organisms, so low salinity is a stressor for them. So this was an experiment we did several years ago, and if you look at the high nutrient here, you see that the salinity, which are the three bars, the white, gray, and black, the salinity has very little effect on the high nitrogen treatments. But if you look at the low nitrogen treatment here, that low salinity, that 15 PSU, 15 parts per thousand, really reduces the ability of this alga to grow. Give it enough nutrients, and it can grow regardless of the low salinity. The result of this high nutrient uptake and ability to tolerate environmental extremes is that, as I said, the ubiquitous shallow systems, as long as light penetrates all the way down to the benthos, and in fact, they're downright prolific in nutrient-rich estuaries. Think about, and Martha touched on this already, is that excessive nutrients can cause shifts in dominance amongst these primary producers. So if you have very low nutrients, like you have on the left side of this axis, this conceptual diagram, you should get dominance in estuaries by phytoplankton, or if the conditions are right, seagrass. But as you increase nutrient loading, you're increasing dominance by green macroalgae. And if you end up going to really, really high nutrient loading, often get another transition to cyanobacteria. So these excess nutrients cause shifts in the dominance of different primary producers. It's just a simple, actually it's a pretty complex conceptual model to sort of tell you, summarize what kinds of things, effects that macroalgae can have on estuarine ecosystems. And so look at the one on the left, and that's a minimally disturbed or a low nutrient system. It's got a very small purple arrow up in the upper left. In places where the nutrient loads are low, the light penetrates all the way down to the benthos, down to the bottom of the estuary. Supports high photosynthesis, can also support high amounts of oxygen in the sediment, nitrogen recycling, some loss due to denitrification, a lot of bioturbiration, a lot of organisms that can bring oxygen down into those healthy sediments. As increased nutrient load to the right-hand side, and then that's the big purple arrow, you can see that you get these big swing rafts of macroalgae that often blocks the light, stops oxygen production at the surface of the ocean, at the surface of the sediments. There's a lot of uptake of oxygen and loss of oxygen in the sediment and in the water, and really can often, as Lauri will show you later, kill off those bioturbators that bring the oxygen down into the sediment. And what happens is you get that acidic or low oxygen level and high sulfite sediments exist right on up almost to the surface of the estuary. That also is what kills those invertebrates that live there. Just to sort of summarize that the main endpoints of concern that we touched upon last slide, nutrients cause poor surface water quality, day fluctuations in oxygen, hypoxia in the sediments, increased bacterial growth, which also increases respiration and draws down oxygen, or poor benthic habitat quality that increased sediment organic matter, is up with sediment anoxia almost to the surface of the sediment, increased pore water sulfide ammonia, all those sorts of things that are toxic to a suite of the organisms that live in the sediment, and that therefore changes foods in supply of food for upper trophic levels, and can result in the critical loss of habitat for fishes, birds, and many types of invertebrates. And as you see in one of the pictures, that was the fish kill we saw in San Diego estuary. So there's lots of literature on the effects of macroalgae, but there's very little that actually try to hone in and characterize the dose response that would be really valuable for endpoint selection. So the instance that we want to keep considering macroalgae is that they have a relatively predictable relationship with nutrients. It is complex. Best example of looking at a loading response situation where macroalgae is responding directly to total nutrient loads is Qualit Bay. They had a big margin ecosystem research project going on there, and they did a really fine set of measurements and experiments to show that total nutrient loads are a really good predictor of biomass in these 3 different subbasins of this estuary with very different loads. So they're able to do real-time comparisons of these 3 subbasins. But like I said, the relationship is really complex, and it's easiest to determine where river sources of nutrients are dominant, which is always the case here in California. And also, what came out of this work is that cofactors play a large role in regulating this response to nutrients. One of we have to deal with here in California is the huge temporal and spatial variability in the blooms. So here's just an example from Carpinteria Marsh up near Santa Barbara. You look at these graphs. One is percent cover, so you can look at either if you like. But the middle head, the head of the estuary, is the one closest to the sun coming into Carpinteria Marsh. The mouth is the one closest to the ocean. You have a real strong pattern of more algae near the river mouth, where the nutrients are coming from. And a real strong pattern of there being winter blooms. You see the white bars and the gray bars are wintertime. And in Carpinteria, at least, those blooms coincide with fertilization of strawberry fields, a direct nutrient source. Another fun fact to know that you'll need to know for later on is the way we typically measure macroalgae is to lay out transects, which is the yellow line you see right here. We randomly lay out transects, and then we choose quadrats, which is the little square box you see with the string with the graph on it. Use these to estimate percent cover, and then also to collect within that quadrat to estimate biomass. So just in summary, things you need to know before you hear the rest of the talks about macroalgae, that I can't stress just they are a natural and beneficial part of all aquatic systems, especially estuaries. Again, too much, often they can exceed thresholds where there's too much. Flavors of macroalgae red, green, and brown, with the red and the green being the ones mostly involved in blooms. Rapid uptake, plasticity and growth form, high tolerance to environmental extremes, and makes them very prolific in large accumulations in anthropogenically disturbed systems, particularly those with high nutrients. As nutrients increase, macroalgae outcompete other primary producers. There are cases where there are really well-documented relationships of macronutrient loads, but the final thing to say is that they are spatially and temporally variable, typically, as I said, estimated by percent cover and biomass.
Okay, so I will transition to the next talk. If there's anyone that if there are any submissions of questions, we can take a minute or two for questions, and if not, we can go ahead and move on. Go ahead and pass the control to me, please. Thank you. All right. So now we're moving on to sort of meat of the presentation, and hopefully the previous two presentations kind of gave you enough of an overview about the NNE, as well as understanding a little bit more about on macroalgae. Now, the two presentations are going to be focusing on the actual work that we did to support, to produce science that would support decision making with respect to thresholds for macroalgae and estuaries. And to kick this off, I'd like to introduce Dr. Lauri Green, who is a postdoc at the Harvard Branch Oceanographic Institute. But Lauri has been working on this issue and actually did her dissertation work on it, as well as a postdoc, a joint postdoc between UCLA and SWRCB. So she's an ecologist, and her research has really been focused on investigating the relationships between macroalgal blooms, habitat quality, and the success of estuarine bird foraging, as sort of essentially linkage to beneficial uses. She received her PhD from UCLA under Peggy Fong, the Department of Ecology and Evolutionary Biology, and she, as I mentioned, is now at Harvard Branch Oceanographic Institute, where she's continuing that work. I should have you pass the control over to Lauri, Eric. Thank you very much. Lauri, mute.
Okay. Hi, the pound key instead of the star key. Hi, everybody. I hope everybody's having a good Halloween. Martha, did you have a slide that you wanted to present before me?
No, we're all set. Keep going.
Okay, we're all set. Okay. So,
oh, I'm sorry. Keep going. There we go.
All right. What I'm going to talk about today is first I'm going to talk a lot about infauna and epifauna, and I want to start by saying focusing on this response variable. I'm going to kind of talk about some of the work that I did as a graduate student under Peggy that actually paved the way for the experiments that I'm going to talk about a little later in the presentation. Excuse me. And I'm going to talk about the methods that we used, highlight some of the key results, and ultimately bring this back to the synthesis of all of this. So, why the focus on infauna and epifauna? Well, first of all, for quite a long time, infauna and epifauna have been a very effective response indicator for things like toxicology and other things. But also they're really, really effective in indicating to us the effect of, say, nutrient pollution on changes in food web structure, chemical cycling. And the reason why they're such a good response indicator is because they're critical to the food web. They're eaten by a lot of organisms. And they're really, really critical in that oxygen penetration that Peggy mentioned and nutrient cycling. Today, I'm going to focus a bit on functional groups. So, for example, I'm going to be mentioning surface deposit feeders quite a bit, suspended feeders, herbivores. The reason I'm talking about these is because these groups in particular are important for grazers. So they're the ones that are grazing, say, phytoplankton, the microphytobenthos, so the benthic algae, macroalgae, and largely they're responsible for taking that carbon from primary producers and moving it up the food chain. So that makes them really important themselves. Also, they're really, really important prey for birds, for fish, for other crustaceans, some of which are, say, commercially important as well as being very ecologically important. Also, these organisms are, as I keep mentioning, important for perturbation and increasing oxygen penetration into the sediments. So I'm going to be discussing some results that compare a little bit between surface deposit feeders. Oh, let me get my pointer. Deposit feeders and subsurface deposit feeders. And I want to begin by saying, there's a striking difference between these two groups because surface deposit feeders tend to live either right at the surface of the sediment, say epibenthic, or at the sediment-water interface. Because of this, they're really readily available to birds of different sizes. You can see my little bird with its little bill, or a larger bird with a larger bill. They're available as prey for fish and crabs and sometimes even stingrays. Surface deposit feeders tend to burrow quite a bit deeper. They're often inaccessible except for maybe the largest birds, the deepest burrowing predators. And also the difference between the two is that often the deposit feeders don't bioturbate as well. So they're not bringing that oxygen-laden water deep enough into sediment, and which affects all kinds of processes. So here's a cartoon, and then also some images that were taken with a sediment profile imagery to highlight the fact that in a healthy or less stressed environment, you're going to have a diversity of all of what I mentioned. You're going to have surface depositors, you're going to have herbivores, you're going to have suspension feeders, you're going to have subsurface deposit feeders. And as a result, you're going to get a nice, deep oxygen penetration, very light-colored sediment, and probably very effective nutrient cycling. To increase the stress in these environments, however, you lose that diversity. You're going to lose some of these key functional groups, which is something that I'm going to talk more about, and your oxygen penetration gets shallower and shallower and shallower. The result is that you have in the final little panel where you have basically an oxic sediment coupled with high levels of sulfide, and very little nutrient cycling may be occurring, and also very little prey availability. This is not the ideal state that we want to be in. So I'm going to talk to you about how some of the results of the experiments link to this. So first, to ask the question about what is the effect of algae on the benthic community. Hull did it back in 1987, and other people have attempted to tackle this question. Typically, the way they've done this, though, is that they'll take algae, they'll put it into like mesh bags or something, and they'll put it on the benthos. In some cases, they're just doing like one single treatment, one abundance. Others did a couple of different treatments and then measured them at pretty far distances, say at 10 weeks or 22 weeks. The difference between these studies and the work that I'm going to present is that one is multiple treatments, and two, I kept the abundances of macroalgae constant over my treatment period, so that we can really identify, what are the effects between this treatment and that treatment. The problem with studies is that often the samples of the macrobenthos may have been taken quite after the algae had decomposed, and we really don't know when the change occurred. So, as a graduate student, I started to tackle this, and I'm going to talk to you about that now. Just let you know that my early work was conducted at Macroalgal, which is located in Southern California. It's a highly eutrophic estuary, but what we did is we constructed cages, as you can see in the cartoon and as well as in the photograph. These cages were 1 meter by 1 meter square plots, lids, and we put different abundances of algae into the cages. I do want to bring up this 0 to 60 treatment. an attempt to have a zero treatment. So the algae was kept out of the cages. They were cleaned. But in this environment with such a high nutrient load, then the algae grew back every 2 weeks. So it didn't turn to be a true zero treatment. But with the way the algae, we found it was about 60 grams of dry weight. So finally, we have these 3 different treatment levels. What we did is we set the cages out, did macroalgae dosing at Benthic samples were taken initially before the treatment set out, and then every 2 weeks for 8 weeks. We also, during weeks 6 to 8, were able to deploy sediment peepers. And for those of you who are not quite familiar with this is a chamber that I'm holding up. It's got wells drilled into it, filled with oxygen-rich water, and they're pushed into the mud. It allows us to collect sediment water and analyze that pore water for a variety of things, including sulfide, which is really what I'm going to talk about. On the slide, I want to point out that we have different graphs, and this first graph is showing polychaetes, which are surface deposit feeders. So it's on the y-axis. On the axis are the different treatments, my 60, 90, and my 510, and each represents a different sampling week. So it actually starts with week 2, goes to week 4, 6, and 8. Because of variability in our initial in those initial samples, we excluded them from this analysis. So all you're seeing are weeks 2, 4, 6, and 8. But what you see here is that the spikies, or the surface deposit feeders, actually fared pretty well in the 60-gram dry weight treatment. And after 4 weeks, they really declined in the 190. And then the 510 treatment, so that first bar is 2 weeks after I put the out, they're virtually eliminated. So deposit feeders really took a hit with these different algae treatments while staying somewhat stable in the 60 treatment. This also affected the diversity, the benthic diversity of the community. And you see again, here's the Shana diversity. And consequently, as I increased the treatment, benthic diversity went down. In this final, this graph, what you can see is the sulfide concentration. So this is sulfide concentration in micromolar on the y-axis. And again, the different treatment levels. And 60 has not that sulfide concentration collected from the pore waters. But start to hit the intermediate and the high levels of macroalgae, the concentration collected was much higher. And, almost record high concentrations collected from these sediments. Respond very well to this loss in surface deposit feeders, and from literature we know that surface deposit feeders tend to be pretty sensitive to sulfide accumulation. So this study told us about, kind of maybe where thresholds might be. We might have causal effect here, mechanism of sulfide accumulation, but in order to make this applicable to water standards, we need to get out of Magoo and see if we could find some commonalities between the results into other estuaries. So we selected 2 estuaries in California. We selected Bodega Bay and Upper Newport Bay. Amongst other things, they respect to climate, hydrology, and sediment characteristics. And we're looking for is to see, can we find a benchmark that's similar between estuaries that vary greatly, also with respect to, say, watershed development. So that will really help us hone in on where the thresholds might be occurring. Out initially, and we did some basic sediment characteristics. We collected cores, and what I point out is that on the left-hand panel, we have the content. And in the 2 sites, in each of we noticed that Bodega had lower organic content in the sediment. Upper Newport Bay had higher organic content. 1% sand, and noticed the reverse was happening. The Bodega typically had higher sand, while Upper Newport Bay had consequently lower sand. So just for a basic measure, we're going to say, yes, there are some differences occurring between these estuaries, and actually between the sites within the estuaries. The kind of protocol was virtually identical to what I used during my PhD work. We had these different cages, exactly the same, 1 meter by 1 meter. We used a higher range of doses to expand knowledge of where the thresholds might be occurring. And we took samples initially and collected Benthic community samples every 2 weeks for 8 weeks. And this is a picture that was actually taken at Upper Newport Bay. A lot of information on this graph, so let me kind of point to some key things. On the y-axis are numbers of surface deposit feeders per meter squared. And each graph represents the different sites. So Bodega 1, Bodega 2, Upper Newport Bay 1, and Upper Newport Bay 2. And also the individual bars representing a different sampling week. So you'll notice that in these graphs, we were able to include the week 0, weeks 2, 4, 6, and 8. The important message to pull out of this is that we noticed a decline in the surface deposit feeder populations at the lowest treatment level that we used. So this approximately 110, 120 grams dry weight for 3 of our 4 sites. And these effects occurred fairly rapidly, within 2 weeks, within 4 weeks, thereabouts. So with our lowest treatment of macroalgae. And we're not presenting those graphs. The similar patterns are actually found with the diversity, the herbivore populations, and suspension feeders. One in Bodega 2 and Upper Newport Bay 1 showed declines with that lowest macroalgae treatment, and Newport Bay 2 didn't show effects. So it begs the question, what makes Upper Newport Bay 2 less responsive to macroalgae? And one hypothesis that we have is that the benthic community composition is a little different, and it may be less responsive. So what we're looking at was the subsurface deposit feeder populations at these different sites. So again, on the y-axis, you have the subsurface deposit feeders per meter squared and different sampling weeks, and we have each of the different sites. And what you'll notice is that the subsurface deposit populations are pretty low. Actually, for the 3 sites that had strong responses to macroalgae, for B2, we actually had quite a high response. We had 8 subsurface deposit feeders that seemed to increase in some of the treatments over time. And this may have affected the total infaunal increase at sites to macroalgae, because as you'll see, you'll compare Bodega 1 and Bodega 2, we saw losses in total infauna. For again, 2 weeks. We have it within 4 weeks at these sites. But in Newport Bay 2, we're seeing these increases. And the reason for this is because subsurface deposit feeders are known to be resistant to, say, anoxia, to sulfide accumulations that occur when the mats build up. So we already have a population that is loaded with resistant taxa. We're not necessarily going to see a strong response of macroalgae. With all these results tell us, well, we were able to obtain the lowest observed benchmark, right, which was that 110 to 120 grams dry weight of macroalgae. So in summary, we did see strong negative effects of both fauna and epifauna at this macroalgal treatment that we used, so this 100 to 120 dry weight. This response was pretty fast, and we saw it within 2 weeks in some cases, within 4 weeks in other cases. And I work as a graduate student. I know that macroalgal mats can persist in location for months at a time. So we can have pretty strong effects of the community very quickly after a bloom occurs. So importantly is that this benchmark abundance was similar for estuaries that really vary quite widely with respect to all those characteristics that I mentioned. So it's actually really helpful in informing where these thresholds might be occurring. The caveat on some of this is that where you have a benthic community may have, say, stress-resistant taxa, such as subsurface deposit feeders, where we may not necessarily see a strong response of macroalgae. I think it goes back to Martha now, right?
But if we have any time for questions, we can take that. And then the other thing I would just really want to just emphasize is that Lauri's work is important because when we talk about the lowest observed kind of benchmark, this is something that, from my opinion, is the first time that someone has documented, really carefully with an experimental study, what number would be, and we can't say that is the sort of tipping point at which the macroalgae is inducing a change in the benthic community. It's the lowest documented effect that is sort of available in the literature thus far. So, if that's helpful for people. So, Eric, we have one question from Laura Sanders. It's not specifically for. Would you like me to try to answer that, or? Okay. So the question is, since there's so much variability spatially and temporally in each estuary, since there is so much variability spatially and temporally, will estuary need its own standard? And so I think that's a great question, Laura. Let me break it apart, because Peggy's comments is that macroalgae is very spatially and temporally variable, and what's really helpful to understand about that is sort of two things. What we mean by that is that it's patchy, and so we're going to use it as an indicator. Then you have to monitor in a way that accounts for that patchiness. It means doing a good job with both kind of sample of the estuary, recognizing that estuaries naturally have these gradients from high nutrients to low nutrients, and that is typically, not always, kind of as you go from your freshwater source out to the sea. And then the other thing is understanding, temporally, it's not always predictable when you're going to have a macroalgal bloom. Some places it may peak in the summer, other places it may peak in wintertime. And so when we monitor and when we develop an assessment framework for macroalgae, that will have to be taken into account. And so then, the other reason why we like to use a combination of biomass and percent cover is to be able to account for that patchiness. And so when you think about kind of objectives or standards, what I'd like to be able to include is information on the magnitude, which is, how is it, like how much do you have, that's the abundance part, extent, which is, the spatial component, and the duration. And so we will be able to understand, and Lauri presented some key results on at what point do we start to see a problem, and that's as early as 2 weeks. And so understanding that, we need to incorporate duration and how long we see the blooms. And the blooms can be variable in the estuary. Some only last a couple weeks, others may last for a month. And so we need to be able to take all of that information into account. So that's, as part one of your question. Part two is, will each estuary need its own standard? And if the standard that you're talking about is nutrients, and it's possible that they may, because, and this kind of comes back to the whole premise of the NNE and why nutrients are different from other contaminants, is that because of those site-specific factors, those site-specific factors may change the equation, the nature of the relationship between nutrients and what's responding to it, in this case, let's say macroalgae. And so the site-specific numbers for nutrients for each estuary may be different. And we're not going to talk about the load response modeling today, but that's part of what that phase of the work would need to be able to address. Okay, so with that, what I'm going to do is, if you have a follow-up question, go ahead and post it. I'm going to keep rolling. Okay, so actually, let me back up for a second. So, so the next part presentation is focusing on another part of the work that we did for these NNE studies, look at experiments. Lauri did all this work, with a lot of people helping her. But so the first part of the presentation was based on experiments. The second part is based on a field survey. It's a very different approach to think about when we get to numbers, and I think the two are very complementary when considered together. Okay, so, and this is the rub, because the bottom line is that when you're doing experiments, you get great data because you get a benchmark. The problem is they're done in specific places under very specific conditions, and you're always going to want to ask, well, how do those conditions represent the rest of the estuaries that we have in the state? And really valid questions. How do we extrapolate these findings across estuaries? And so our approach to this, recognizing that the state has a limited money in which we can do science to try to support decision making, is to a field survey in which we want to be able to use the idea of sediment oxygen penetration, what I'm going to refer to is the redox potential discontinuity. Let me grab my, so that's a very long word. I'm going to abbreviate it by calling it ARPD. And what it is the depth of oxygen penetration in the sediment. What happens is that if you have a lot of infauna that are moving around, especially large ones, they create burrows, they pull water out of those burrows, they're increasing the oxygen penetration of the sediment, and that pushes down or presses the depth to sulfate reduction in particular. And so that's to maintain this healthy oxygenated environment, mildly oxic, I should say, in which these infauna can thrive. And what happens is that, and this conceptual model is showing this gradient from kind of really good condition sediment on one side, and as you move over to the right, it's becoming increasingly eutrophic. What happens is you see this depth, this RPD, this depth to the oxygen penetration begin to become more and more shallow, and eventually it zeroes out, and at which point it is essentially becoming azoic. What happens is that we can take advantage of this technology called a sediment profile camera, grab imagery to document the depth of oxygen penetration in the sediment, and we can measure the depth of the ARPD. And so what happens is we take this camera, we push it down into the sediments, and then we're basically measuring and integrating over the face of the camera what is the depth to that ARPD discontinuity layer. Okay, so sort of a really helpful way of doing this, because what we can do now is go out and survey lots and lots of estuaries, or at least as much as we can, for the resources that we have available, and then try to understand how this ARPD varies as a function of algal biomass and other variables. And so what we did was a survey during 2010, 16 sites in 8 estuaries. And at each of the sites, what we would do is go out and measure a suite of parameters in 20 plots along a single transect. And what we're measuring is, first we'll go ahead and measure the algal biomass in percent cover. Then we'll take the sediment camera and take a image of the sediment so that we use that to go back to the lab and measure ARPD. And then we're also grabbing a sediment, sufficient sediment sample for sediment percent organic carbon, nitrogen, and percent fines. So what I want to be able to convey is that these 8 estuaries were chosen in order to capture a diversity of California estuaries, including enclosed bays as well as lagoons and river estuaries. And they also had a very large difference in size. And the question is, well, given all this variability, can we see any relationships between algal biomass and RPD, this indicator of the habitat quality? Okay, I'm going to use this graphic to give you a sense of how to think about the relationship between algal biomass and ARPD. And what I'm showing you here is ARPD in centimeters, on the y-axis, and then algal biomass in grams by weight per meter squared on the x-axis. And then the color represents the algal type. Peggy mentioned that there's several flavors of macroalgae. We saw mostly reds and greens. and I also have shown you where there was no algae taken. And then the symbol shape represents the different estuaries, and what you can see is that, among those estuaries, we have a pretty good spread of data in terms of what we saw. In general, what I want to convey to you first is that there are a lot of things in these estuaries that are controlling ARPD, not just macroalgae, not just sediment organic carbon, the hydrology, the presence of toxic contaminants. It's really important to understand that that the data are essentially showing you that it's put at low levels of biomass, you can get a pretty large range of ARPD, but that what happens, and this is sort of the first way that we started thinking about how to use this data, is that some level of biomass of macroalgae, it seems that biomass begins to override other variables and really bring that ARPD down to zero. Remember when I said when you shallow up the ARPD, that's essentially starting to reflect the fact that your sediments have gone azoic, which means it's functionally not dead, but there's very little benthic fauna available to bioturbate there. So this is conceptually how we can look at the data. And what we want to be able to do is, and this is, I think, an important advance that the freshwater literature can provide to how we think about thresholds, is we want to come up with a really unbiased way of identifying those thresholds. And we were able to, through the, we had some great resources from EPA ORD. Dr. Naomi Dietenbeck was a statistician on this project, and she's done a lot of work on this in freshwater systems. And she helped us understand that we can use two different types of statistical methods to identify these thresholds. A classification and regression tree, CART analysis to identify step thresholds, essentially can help us identify what would be considered reference versus non-reference. And secondly, with respect to this exhaustion threshold, she used something called piecewise regression to write slope thresholds. And so what you're doing there is you're saying, at what point do you get a break in the slope where this essentially bottoms out to zero? And so using statistical methods to identify and also identify those thresholds and to be able to place confidence intervals around those thresholds. That's really helpful to get some of uncertainty. Okay, so start with the reference versus non-reference. So these are the step thresholds. Essentially, what we were able to identify is that roughly algal biomass in the range of 2 to 16 weight per meter squared was defined as a reference envelope based on ARPD. You can see that this the mean value is 14, and then essentially, the range of the confidence interval represents the envelope in terms of what we would consider to be reference. And this is interesting because this is the first time we were able to find that someone actually defined what reference would be for macroalgae in estuaries. This is more common for what they do in streams. It's less common for estuaries. We collected percent organic carbon in sediments, and we are also able to use that same data to find what would be reference for percent of carbon or percent organic. It's essentially a measure of organic matter in the sediments. That value ranged from 0.4 to 0.5% organic carbon by weight. Okay, I talked about the reference envelope then. How about for the exhaustion threshold? And so what we're seeing is that using the piecewise regression methods, we actually were able to identify for macroalgae a range of 175 to 190 grams weight per meter squared as the 5th percentile of that slope. And if we think about what that means, and I use this graphic to explain it, what's happening is that your slope coming down, and then your bottoms out. Because for this particular modeling exercise, we used the slope only, our no-break model was the best fit for macroalgal biomass. The fifth percentile represents essentially this point close to where it breaks. So if we use the median, it would be in the middle, and the 95th would be out here. So we're saying that the 5th percentile represents a conservative estimate for where this threshold essentially lies, where this exhaustion threshold lies. Likewise, with percent organic carbon, unlike the slope-only model for macroalgal biomass, the slope break model was the best fit for percent organic carbon. And so what we're essentially seeing is a 1.1 organic carbon defined as the exhaustion threshold based on that site-rich data. So where Lauri left off, she said, all right, well, this study, these are field experiments, identify this lowest observed effect benchmark at 120. Now we're using the camera survey, this Benthic camera survey, to say, now we see that we have a reference envelope somewhere between 2 to 16 dry weight per meter squared, and then an exhaustion threshold around 175 to 190 grams dry weight per meter squared. Okay, so this is what this new data and these studies supported by the State Water Board are bringing forth. And so what's really helpful to understand then, actually I'm going to, I forgot, I have 2 more slides before I get to the punchline. The other thing that's really helpful to understand about macroalgae and sediment organic matter is that there's a very, very strong feedback loop between macroalgal biomass and sediment organic matter content. Essentially what happens is that the macroalgae are sitting on top of the mat. So any sediment nutrients that are available sediments, the macroalgae is very, very good at actually taking them up and producing biomass. And when the algae senesces and dies, oftentimes that algal biomass stays and gets reincorporated back into the sediments, and so you have this very strong feedback loop. Interesting is if you look at the relationship between percent organic carbon and biomass, what you see is that after a certain, when you hit some sort of peak in sediment organic carbon, it looks like, particularly in areas around greater than around 2% organic carbon, that you really get biomass under 100 grams dry weight per meter squared. And so this is reflective of the fact that when you have very, very enriched carbon in your sediments, you're going to be fueling macroalgal blooms, and that's an additional source for what fueling these blooms into what's coming in through the water column. One thing I wanted to cover with you guys, no pun intended, Peggy, that we had biomass and percent cover is how we as these how we estimate the abundance of macroalgae. And what's important to establish is, first of all, we didn't see any relationship between percent cover with ARPD. But what we like to understand about percent cover is that it may have potential as a screening tool, and this is the result of a quantile regression where we look at the relationship between percent cover and biomass. And what you can see is that at very low biomass, you essentially are, I would say, I'm sorry, I'm going to back this up, a very low cover, you typically have low biomass. And so the way that we can use this is potentially as a screening variable, that less than something like 30% cover, there's only 5% of the plots that actually exceed a biomass of 14, which is roughly what the top reference envelope is. And so maybe that as we think about how we monitor macroalgae, when you have low percent cover, generally you're not likely to a position where you're causing a problem, in which case the analysis of biomass may not be necessary. So these, lots of uses for how we will be thinking about using these data and not just for thresholds. So to wrap up with the sediment profile imagery survey with respect to findings, first I think that this project established a reference envelope and exhaustion thresholds for macroalgal biomass and percent organic carbon. The reference envelope for biomass was 2 to 16 grams per meter squared. Organic carbon, it was a range of 0.4 to 0.5. We had a resistance threshold of about 175 to 190, and 1.1% organic carbon. And again, mentioning that we understand that these variables are very strongly related and indicative of a feedback loop that has been very, very well established in the literature. Fong and her students and a lot of other scientists have really explained this conceptually, and there's a great deal of literature that documents this. And there's no relationship between ARPD and cover, but it's possible to use percent cover as a screening tool, and we'll be thinking about that as we move into the development of the assessment framework. Okay, what I'd like to do is we could go ahead and take questions now, except that what I think I've only got a couple more slides, and so what I'd like to do is wrap those up and spend the rest of the time answering your questions. Okay. What we've done through this work, then, as I said, we've established this lowest observed effect benchmark, a documented reference envelope, and then this exhaustion threshold with respect to ARPD. And as well, all right, that actually this is a tremendous advance, but over previous work, I think if you think about how people would start to use macroalgae as an indicator, it's been largely used either qualitatively or as folks have made decisions about how to assess macroalgae and how bad it is based on best professional judgment. And this is really helpful. But I think the issue that the State Water Board will still need to deal with is that these studies by themselves do not inform two things. It doesn't inform the effect level, as well as, so some sort of effect level or benchmark, as well as the resistance threshold. What is the point at which things start going bad? And at this point, based on these data alone, we do not know the distance, essentially, between this lowest observed effect benchmark and resistance threshold. So the question we would want to ask is, are there any additional studies that can help to inform that? When we think about using additional studies, the problem is that that we get into try and interpret other folks' data where they've used different methodologies. Europeans like to do things with respect to wet weight, biomass, in which you're not cleaning all the sediment out of it. There's a lot of difficulty in how we would use other folks' data, and so in order to help the State Board and the stakeholders understand, think about these other studies, we like to assign levels of confidence. And so when we ask the question, can other studies fill in the gaps in the thresholds, there are 4 studies in particular that I think that we've decided that could be considered, and that we've assigned some level of confidence from high to low and how they should be, and how those results should be treated. So first of all, about this idea of an exhaustion threshold, Lauri's PhD dissertation that she presented to you was a great experiment. It was a really high quality experiment with controls, treatment, replicates, consistent application of biomass, and that's important. Not a single dose, let it go, because what you're really trying to do is if you need to, under the threshold, you need to maintain the biomass over time. And she was able to document that at 190 grams per meter squared, she saw severe effects, and with sort of a correlate or at least causal data documenting the very high pore water sulfides. And so this is really important, and sort of supporting data, have a high level of confidence in data. Moving down on the scale, Francesca Bonetta did a field survey with a Benthic camera in a single estuary. This has been in Venice Lagoon, and what she documented, so this is like our camera survey, but in one estuary. She documented essentially the loss of these large burrowing bivalves and tube worms, what she's called Benthic, stage 3 Benthic colonizers, at about at 90 grams dry weight per meter squared. That was a big study, although it was not necessarily intended to address thresholds. She cites a threshold The threshold was not derived statistically, it was essentially eyeballed, and so that's important to think about that as well. Then moving down to the studies that I would kind of put in the of low confidence, and there were two. The study by Cordosa et al. In 2004. It was a field experiment with a single application. They documented no adverse effects at 30 grams dry weight per meter squared, even though they reported those results in weight. That's one uncertainty. They also did a single application, so we really are not confident when they say something's 300 translated in 30 by a simple trick between wet and dry weight. That actually represents 30 in the way that we measured it, and so that should be taken with great caution in how you interpret it. And then Lauri Green's field experiment, Lauri said, she had a 60-gram dry per meter squared treatment, but essentially what that constituted was originally supposed to be a control, and she was removing algae at each week interval that she was maintaining these things. And what happened is that actually growing back, and so for what we went out to measure it again, it actually constituted a 60-gram treatment, actually varied between 0 and 60 grams. They also found no effect. The problem, again, with that is that the biomass was not constant. Even though the study was a great study, the biomass was not constant, so we don't know sure what that number means. Well, so then if you think about it, these studies can shed light on the information gaps, in particular, high confidence in the severe adverse effects, really, I think, sorting this exhaustion threshold that we documented with the camera survey. This resist threshold represents approximately where the Bona paper would put the 90 grams per weight, 90 grams dry weight per meter squared. So this is sort of medium confidence, and then the low effect, let's say that's approximately 30, but then we have very, very low confidence in numbers, and this is still an area that I think additional research needs to be done. Ultimately, State Water Board is not necessarily intending to do additional studies to identify, to further this work. They need to move forward and make decisions, and so it's a policy decision ultimately on what science they will choose to use to make those policy decisions. And I think that's essentially what you guys should be commenting on of thinking about as you move into the next phase. Okay, so what are the steps? And so what we've done, if you kind of go back for a second, so now we've synthesized this information on thresholds. And there's a lot of things that need to be considered as well, but essentially the next step is to develop a macroalgal assessment framework. And I mentioned there's 3 components, one for seagrass, one for tidal flats, and subtidal habitat in open estuaries, and the third is a macroalgal component for closed estuaries. What we're next on is this for intertidal flats, shallow subtidal unvegetated habitat. And the way we're thinking about it is essentially by looking at what others have already done, and there's really some great work that's the European Union, what's been proposed for the Water Framework Directive by Scale. Essentially what they're doing is they're using a combination of biomass and cover to estuaries from very high condition to low condition. And you can see they've made judgments on what the how to make decisions on where the categories are, but those decisions were actually informed by best professional judgment. And so what we're considering, and we would be interested in feedback as we go along, essentially trying to modify this framework and using our information on thresholds to think about how you would adapt this framework to work for California estuaries. Ultimately, and I want to reinforce this, how you would, the decisions on, essentially if you think about how this translates back to the N&E, if we have the categories of what is sustained uses, 3, which is definitely impairing them, and then 2 is sort of in the middle, the state board would need to make a decision on what those regulatory thresholds would be. And the assessment framework would essentially help to support that application and for applied in different different programs. Okay, so I want to take a moment and acknowledge our co-authors. I mentioned Amy Dietenbeck, who was our statistician, tremendous there, Giancarlo Sacchetti, both from EPRD, the National Health Environmental Effects Lab, and Narragansett. Giancarlo was our expert use of the sediment camera, funding provided by the State Water Board. We had lots of help on field work, as well as taxonomic quality assurance. A note, I have this as acknowledgments. These are all the references we cited, particularly with respect to the thresholds. I want you folks to be able to have access to those papers if you wish. And I wrecked the title on the final version of this. And that will take any final questions or comments.
You can ask your questions by unmuting your phone, press *6. It can also be submitted via chat.
A question I was reading from Chris was bacteria observed or measured in these field surveys at the 8 estuaries? Not measure cyanobacteria specifically. We did, however, measure benthic microalgae, of which cyanobacteria would be considered to be a component. And not report on that data, but that's part of what we actually analyzed as part of this study. A question from Dinah Engel that says, are you saying that stakeholders can obtain copies of the manuscripts in review? And the answer to that question is yes, and what I'm going to do is I'm going to post those manuscripts on the NNE website. It's a website that I created to distribute those documents to stakeholders. It's not something that we normally do, is send out stuff that is in review, but I think it's really helpful to get your feedback on things. And since both of the manuscripts have been submitted, and we typically take about a month or two to get the final reviews back from the editors, we're happy to have any feedback that you guys are willing to give to us. Yes?
Can you add that web link to your presentation, which I'll web post?
Yes, I will.
Thank you.
Mm-hmm.
Hey, Martha. This is Christine Jubb. I had a question, and I don't know if it would be something for you or Lauri, but do you get any idea of whether or not the surface deposit feeders or the subsurface deposit feeders over time can develop a little bit more resistance to this type of stress impact in the environment, so that over time, I was just thinking about that Upper Newport Bay 2 site and just wondering if organisms within that environment have been exposed over a longer period of time to this increasing sort of eutrophication process that has made them a little bit more resistant or more tolerant. Do we have any sort of information from literature on whether or not that is possible?
Want to take that, or do you want me to try to answer it? I can jump in, I think I answered that pretty well, Chris, because we've actually done a lot of work in Newport Bay, and we have with a lot of different types of studies. And the interesting thing about Newport Bay is that, at least where the Newport Bay 2 site is that that's an area that has a lot of problems, not just eutrophication, also toxic contaminants. And so what's really helpful to understand is that site is really not a great site in the sense that it already has been adversely affected by a variety of stressors and has a low kind of benthic habitat quality as a result of it. The way I would think about, kind of getting back to your question, can surface deposit feeders acclimate over time? I think that generally, even though I'm not a benthic invertebrate ecologist, these people have really reliably used benthic vertebrates with respect to community composition, their abundance and their diversity, make judgments about the quality, the benthic habitat quality of estuaries, and we rely on that information because we know that that adaptation is probably not happening at a rate, if it is happening, that really is going to bias our results. And there's probably a variety of mechanisms behind that, and I'm not the authority on that. But ultimately, I think the way you should think about this is that this comes back in place for how we would use organic carbon and what's the nexus between N&E and things like sediment quality objectives. You may have sediments that are enriched with respect to carbon, but for whatever reason, at that particular time you're going out, you're not seeing macroalgal bloom. And it's possible that although currently the quality objectives are not really being used to assess eutrophication, it could be in the future that additional indicators developed would be able to show data in a sense that if you're doing a sediment quality assessment and you come up with very, very high organic matter content, that it's possible that may essentially become a referral for how to think about that system with respect to with dense or organic matter loading. So that's a longer answer, and I've kind of expanded on your question, but I think that's the way I think it's helpful for us to think about it.
I apologize. I guess I hadn't unmuted my phone.
That's okay.
So one thing I do want to point out is that the surface deposit feeder populations that we observed in Bodega were different than those in Upper Newport Bay. And the species that we did come across in Upper Newport Bay are those that are more resistant, not entirely resistant, because surface deposit feeders are definitely more sensitive to anoxia and sulfide. There's a striking difference between the community composition of these two sites. And so they're the ones that were there in Newport Bay are probably more stress tolerant than the ones that we observed, because most of what we observed up in Bodega were, say, a lot of small crustaceans and so on and so forth, which may be more sensitive to these stressors. So.
Do we have questions? Any more that we haven't covered on the chat? If anyone wants to unmute their phone and ask it, feel free.
Hello?
Hi.
Oh, hi. Hey, Martha, this is Diana Engel. I was typing something out, and I was afraid I wasn't going to get it typed for you either. So this is my question. A lot of these estuaries have rafting algae rather than this attached benthic form, or periodically that occurs. So effects of the floating algae have to be quite different than the attached in terms of this, the benthic macro infauna and smaller infauna. So what kind of steps are going to be taken to address whether they need separate thresholds? How do we know what levels are harmful?
Martha, you need to jump in on that one?
Yes,
this is Peggy Fong. So as I mentioned when I tried to give you the background, when you have the really harmful blooms of algae, they tend to be the big floating rafts. The experiments that we did were all with the big floating rafts. That's what we were manipulating. And Lauri built her cages tough to allow those rafts to float up and down with the tide, because you're sort of forcing an unnatural situation if you turn down and beg them to the bottom, because they do float around. The question is, I'm also thinking, well, they're rafting around, so they're here one day and they're gone the next day. That's why we have two things to think about in that way. One is that Lauri did a survey and found that in many areas the rafts had there's deposition areas, right? And the rafts had quite a bit of fidelity to a spot, and in fact she found that they lasted 8+ weeks on a particular spot, which is the host doing this 8-week experiment, right? We know that it's just on one spot at least that long in many, many places. But the other thing is when she showed you this, she showed you the time series. So we can tell you what happens after 4 weeks, 6 weeks, 8 weeks. So if a mat lasts for 2 weeks, we tell you what the response of the benthic community would be. And in fact, in most places, under most circumstances, for most types of invertebrates, the response was very quick and usually within 2 weeks.
So the notion of like screening tools with percent cover or the application of these experimental results to define thresholds, is there a depth cutoff? Because I'm just thinking, in closed river mouths or some of these subtidal habitats, are we kind of not applying it to those? Because in that case, I would imagine these rafts are not depositing and being refloated.
Mark will have a better answer.
Can I answer that?
Yes.
So the way if we kind of cock the way I want you guys to think about this, and actually let me see if I can pull up one more before we talk about macroalgal and what we just said is okay. We're presenting science that would help support this intertidal and shallow subtidal vegetated application. And then, for closed estuaries, we're going to need to take a different approach, and that's in part because what happens, and I just, kind of showed you a visual, and got to that. The way MAC is manifesting itself is a little bit different here, and I'm not going to come back to that in a second. But for these open estuaries where it's essentially subtidal, and in intertidal, what happens is that we are using the intertidal area as an index area for the habitat, and so and this is something that we're happy to hear stakeholder comment on, because it's a double sword and it can cut both ways. It's much easier to monitor algae in intertidal habitat, and much more difficult and logistically complicated and more costly, therefore, to monitor it in the subtidal. Most of the places that have subtidal habitat are going to have an index, are going to have this mudflat where you can measure as an index, and we can make this relationship or correlation very strongly between the benthic fauna on those flats, and that also is the same issue with subtidal habitat with macroalgae. And in fact, some of the work that's been done previously has really documented the strong relationship between macroalgal biomass, not in terms of thresholds per se, but benthic fauna in subtidal habitat as well. And so using the intertidal habitat as an index area is a pretty good way to go. The problem is that ultimately we need to understand what's the relationship between washes up or sort of essentially presents itself on the intertidal flat versus what's in the subtidal habitat, and that's the way the sword is coming back on us, is that we still need to have a understanding of that, and there's actually some work that's going to be done over this year in an area in Southern California to start to quantify that. That being said, essentially what's being proposed as a part of the NNE is that the assessment would be on the intertidal flat because it's cheaper, and you're impacting those mud flats are very, very important in terms of the aquatic life use support. They support migratory birds. You have a lot of fish that can feed on them when they're flooded as well, and so it's not a stretch for how you think about that. Okay, so I said, but this is different from these closed estuaries, and the reason why we're talking about potentially a different set of thresholds for these closed estuaries is because when the estuary is closed, it fills up, and even though you could potentially have that same conceptual model that Peggy showed, where you have macroalgae, it shades out the benthic primary producers in these closed estuaries, it's causing deep problems. Ultimately, the strong connection between macroalgae, benthic fauna in those estuaries is it becomes more tenuous, and the reason for that is that when you close those estuaries, the salinity drastically changes, and you to have a less rich benthic faunal community, and so and really we need to have more data, and it's not presently there is we need to have a better understanding of the relationship between macroalgae and pelagic invertebrates. And really, those data are not there. And so what we're proposing to do as a starting point is to actually do a reference study in those closed estuaries to get a handle on, one, how do we really measure algae well? And then number two, what is that reference envelope going to look like? And then we're going to have to essentially use best professional judgment unless we're able to the study, and that's not currently being funded by the State Water Board, for how we relate pelagic invertebrates to macroalgae. So that work is yet to be done. This Water Board is not going to be able to fund it because of funding restrictions, and so if we're not able to come up with a way of doing it, they may be forced to make decisions in absence of it. Okay, so really long explanation for your question, but I wanted to make sure it's helpful to put the question. It was a great question into context for how we would think about it.
I had some additional questions submitted via chat.
Okay. All right. So this is, I'm seeing one from Kurt Oling to everyone. The report works appear to illustrate tolerable levels of algae. Is envisioned as the estuary health assessment method. So how will it be correlated to allowable nutrient levels? Okay. So could you clarify when you say the reported works? Could you clarify what you're referring to?
Just all the theme that seemed to come across in the works that were reported today.
Okay, so there are tolerable levels of algae, yes, and so I think that's a message, is that macroalgae is not inherently bad. It's a natural part of the system. There are not only tolerable levels, there are good levels, and maybe reference range begins to help us understand what those levels are. In time, we may be better informed what is essentially what we would call a no effect, where you're getting an enhancement from macroalgae. But then the idea is that we're going to be using some of this information to help to translate, and I didn't really know about how the State Water Board's concept of beneficial use risk categories, but there are essentially three. One is your sustained uses. You're definitely in the good zone. Three is where you're absolutely impairing them. And then I recognize there's sort of this intermediate category where it's less clear, recognizing the considerable uncertainty in how you're trying to manage for nutrients. And so from that perspective, what they want to do is look at the studies that help them pick numbers. There's thresholds between 1 and 2 and 2 and 3. In some perspective, yes. These data will help to inform those thresholds. And then part of your question was how these levels be correlated to, or how macroalgae be correlated to allowable nutrient levels. And so that's a familiar question. That's part of the phase 2 work. We hadn't funded to do at work at this point, although we have some early data that's helping form the thought process for how to do that. And what I'm referring to is there was a study, conducted in Southern California under the BITE Regional Monitoring Program, and it was a survey of 7 sites and 25 estuaries where we looked at the relationship between river nutrient loads, estrogen, surface water, nutrient concentrations, algae, and phytoplankton. So we're essentially looking at the relationship between load, concentrations, and response. And so that report is just being published. We also just submitted, we're actually just about a week away from submitting some of the papers as well on that. So I would invite, and what I'll do is I will add the link for that report to the education survey report. That's I think it may be actually out already. I'll have to check so that you can go and look at what that relationship looks like from the perspective of so a statistical relationship, R squared is, it was definitely a significant relationship between loads, concentrations, and macroalgae, but the R squared or the that was very high. It was around 0.25, and it got up a little higher when you considered things like residence time and hydrology. If that answers your question, I'm happy to follow up with you if you'd like more information. And we're coming
Thank you.
We're coming on to 11:20.
And Mark, could you ask Laura Sanders' question? It's been answered via the chat box, but
really good question. Do we know how long it takes the benthic invertebrate community to recover from partial or fully anoxic conditions? Answer that.
Yeah, sorry, I switched to now. Okay, so the way to answer this question is essentially, it's definitely going to be species specific, but I want to preface this by saying I don't know of that many networks that have addressed this effectively. Some have tried to do it, but their sampling design was not well contrived. So, but what we can say is that stress-tolerant species are going to recover pretty fast. They tend to have a pretty high production rate, and if they've gotten to the point, if the system has gotten to the point of anoxia, as Peggy was trying to also answer, the system is very stressed, and you're not going to recover. The system is going to recover very quickly from that stress state. Only the organisms that can persist in anoxic or hypoxic state are going to be these subsurface deposit feeders, or maybe the stress-tolerant surface deposit feeders. Those would be like early colonizers and stuff. But to ask, when do the suspension feed come back? When do the herbivores come back? When do the predators come back? The answer we really don't know, and we'd have to have a dramatic recovery of the system in order to understand that.
I agree with your answer, and the only thing I would kind of add is that it's really helpful to think about. It's actually, if you read the camera survey paper that we submitted, we talk about this. You would think about that conceptually, that if you already have high sediment organic matter, totally agree with Lauri, it's going to take a while for the system to recover and the benthic fauna to recover. If you have low organic matter content and for whatever reason, maybe a storm, and you manage to have rafted your macroalgae up into a place where the benthic habitat quality was good, it's conceivable that when you remove that organic matter from it, washes out, the recovery may be fairly quick as you have not enriched sediments, and if you get recruitment in from areas nearby, that recovery could be quick, although at this point, it's not clear that anyone's really done work to understand that very well. So, yeah, but check out in the benthic camera survey paper, it talks about this concept of how we may want to use sediment organic matter content as a secondary indicator to make sure that when we do assessments with macroalgae, we are not getting those places where it's just rafted up temporarily. Okay, questions? Well, I personally would like to thank you all for attending. I really appreciate the fact that we're spending a lot of time going over the science, and we're recording this so that people have the opportunity to go back and listen to it. So there's a lot of technical details, and so I appreciate you guys hanging in there with us. Please feel free to email me or any of the co-authors here, or Eric, if you guys have any follow-up questions, we'll try to post the answer to those questions. Thank you very much.